Hello. A lot of energy here. Must have been a good weekend. Everybody's ready for the exam? No? Okay. So I will do, like I said last time, I'll do a review session after class, at the end of the class period today. What I'm planning to do today is I'm going to go through and I'm going to finish up, didn't get my stuff together here. I'm going to finish up glycolysis, and I'm going to do gluconeogenesis, and then we will stop a material for the exam. So to show you where that all is, we will go through the last of the carbohydrates, which is just a little bit of stuff down here that I haven't talked about. I'm sorry, not carbohydrates, glycolysis, sorry. A little bit of the glycolysis that I haven't talked about, which is right here. There's a little bit of stuff down here. Then I'm going to talk about gluconeogenesis, which is right there, and then I'm going to stop right there. So that's where the material for the exam will stop, is at the end of gluconeogenesis. Okay. Well, that said, let's finish talking about glycolysis. So glycolysis, I will remind you, is the pathway we talked about last time involving the breakdown of glucose. It's a 10-step process. I gave you some enzymes that I thought that you should know the names of. And 3 of those 5 enzymes that I gave you were enzymes that are important in regulating or controlling the pathway. Now, I've mentioned to you earlier that controlling pathways is important because pathways result in either the breakdown or synthesis of molecules. They either produce energy or require energy, and the cell's needs have to be met. So, for example, if the cell has a need for energy and it is making molecules which require energy, that would be counter to the interest of the cell. So being able to control pathways is absolutely critical for the cell to be able to respond to the environment in which it's in most effectively. Very, very important. So every pathway that we will deal with will have some consideration about regulation. Regulation will be important because, again, if cells don't regulate properly, that cell isn't going to be around for very long. Well, here's a consideration for regulation. This is a nice schematic showing the pathway. It shows only one of the 2 halves of the fork that you saw last time, but you remember that if we just double this, that we've got the rest of glycolysis going on. Now, you'll notice that there's a couple places where we see inhibition, et cetera, et cetera. Okay? So I'm going to start at the very top and talk about hexokinase. Hexokinase is the first regulated enzyme, and not surprisingly, the first enzyme in a pathway is a very convenient place to regulate a pathway, because if I shut off the first enzyme, I shut off the whole pathway. This is the feedback inhibition mechanism that we talked about before, except for it turns out that hexokinase is not inhibited by feedback inhibition. Okay? It is the first enzyme, but it's inhibited by another mechanism, and the mechanism is confusing enough that we're not going to talk about it, unless you guys want. No. Okay. I knew what the answer to that was before I asked the question. Okay. But you should know that hexokinase is a regulated enzyme. Okay. The second enzyme that's regulated is the one that I described as being the most important regulatory enzyme, and that's the phosphofructokinase, or the PFK. That's the one that catalyzes this reaction going from fructose 6-phosphate to fructose 1,6-bisphosphate. This enzyme is the most important regulatory enzyme in glycolysis because it's very, very sensitive to a couple of things. It's also an enzyme that does what I call reading the energy state of the cell. It reads the energy state of the cell. Well, how do you read the energy state of a cell? Well, a very simple thing to do would be to be able to detect how much ATP and how much AMP that a cell has. AMP is a measure of very low energy. ATP, of course, is a measure of high energy. Well, it turns out that PFK actually responds to both. It responds to both ATP and to AMP. And I want you to think about this not in terms of something to memorize, but think about it in terms of, well, that just makes sense. Here's how it goes. It turns out that Pfk is inhibited by ATP. ATP, I've already told you, is a measure of high energy. The more ATP there is, the higher the energy state is of the cell. Do you want to be burning glucose when the cell has plenty of energy? The other question is, do you want to be lighting your fireplace when it's July? You don't want to be doing this, right? So ATP concentration being high turns this enzyme off. It's very logical. By contrast, in wintertime, it's very cold. And in wintertime, you want to burn your fireplace so that you get heat, so that you stay warm. Right? AMP is a measure of low energy. When the cell is low in energy, it wants to make more energy, just like you want more heat, the cell wants more energy. AMP turns the enzyme on. Okay? So these make very, very good sense. Now there's a third molecule up here that sounds like a molecule in glycolysis, but it's not. It's called F26 Bp. It sounds like F16 Bp, but of course it's not F16 Bp, it's F26 Bp. F26 Bp turns out to be extremely sensitive way to activate the enzyme. Very tiny amounts of F26 Bp will turn this enzyme on. Now, unfortunately, F26 Bp is not really an indicator of high or low energy, so you can't use the logic that I gave you earlier about ATP and AMP. The way in which and the reasons for which F26 Bp are made are a little complicated as well, so we won't talk about them. But I will tell you briefly that they respond that the synthesis of F26 Bp is related to hormonal stimulation of the cell. Hormonal stimulation of the cell can stimulate the production of F26 Bp and therefore the activation of Pfk. Now you might wonder, why do we regulate the third enzyme in the pathway instead of putting everything on the first enzyme in the pathway? And the answer to that question is that both glucose 6-phosphate and fructose 6-phosphate are useful for other pathways in the cell. So this provides a way of shutting down glycolysis without shutting down the production of these two molecules that are used in other pathways. Okay? So this fine level of control here gives the ability for the cell to keep other pathways going even though they've shut down glycolysis. The third regulation here is not really a regulation of an enzyme. It is in a sense. It's a regulation of a reaction. Well, the reaction occurs on the enzyme, right? What is required for a reaction? What's required for a reaction are substrates. If we don't have substrates, we won't have a reaction. And if you remember, when I talked about NAD and NADH, I said the cell had limiting amounts of them. The cell has to recycle them. If the cell has all of its NAD in the NADH form, there won't be any NAD for this reaction to occur because this reaction requires NAD. So the cell has to make sure that it's got sufficient quantities of NAD to keep this reaction right here going. And this is why fermentation occurs. I'm going to talk about fermentation in a little bit. But this is why fermentation happens. Because the cell has a very important interest in keeping this reaction going. If all it has is NADH, this reaction will not go, and glycolysis will not occur. The third enzyme in the pathway is a really unusual one. It's the last enzyme in the process. Pyruvate kinase, which is right here. Oh, by the way, this is G3PDH, if you're wondering what the enzyme was. the one I required you to know. The enzyme down at the bottom is Pyruvate Kinase. And it's regulated by a couple of things. First of all, it's inhibited by ATP. What's ATP a measure of? ATP is a measure of high energy. Again, we don't want the pathway going. Shuts it off. AMP turns it on. And AMP is a molecule that's a measure of low energy. And again, we want glycolysis going if energy is low. So this makes sense too. But now this third guy here is really interesting and cool. Okay? I'll tell you why it's interesting and cool. F26 Bp is, in fact, an intermediate in the pathway. There it is right there. This intermediate in the pathway is doing something I call feed-forward activation. You've heard about feedback inhibition. This is the opposite. This is feed-forward activation. And I'm going to tell you why it's cool and why it's important. Okay? Let's imagine, if you will, that I am out and I am, actually maybe I'm sitting here in class like you are taking notes, and all of a sudden the fire alarm goes off, and all of a sudden this big wall of fire comes from right here. What are you going to do? Well, you're going to get up, you're going to go running as fast as you can, as far away as you can, which means you're going to need energy, right? Energy in the form of ATP. When you take off running, you very, very quickly burn the ATP that's stored in your muscles. That doesn't take very long at all. Well, now all of a sudden you've got this deficit of ATP, and you need to make some more. Well, the quickest way you can make ATP in the body is by burning sugar. Here's the pathway that's going to do it. Okay? So you've just taken off, you've gone racing away, and now you need energy. Well, your body will respond, as we'll talk about tomorrow. Your body will respond with adrenaline. And adrenaline will cause your blood supply to load up with glucose. Your blood glucose levels will go really high. This is how adrenaline works. Giving all kinds of glucose to your muscles so glucose can burn it, so the muscles can burn it, right? But there's a problem. And if you remember last time when I talked about the aldolase reaction, I said there was a problem with the aldolase reaction. What was the problem with the aldolase reaction? The energy barrier, right? Energy barrier. Well, now I've got this process started, and all of a sudden it comes down to here, and this enzyme is saying, I'm sorry, we don't have enough energy. We don't have enough material to get around this problem. What's going to happen? Well, if you don't have something working for you, you're going to collapse. All right? If this enzyme is not working, what's going to happen to the concentration of F16 Bp? Up, right? F16 Bp concentration is going to go up. When F16 Bp concentration goes up, what's going to happen to this enzyme down here? Pyruvate kinase is going to be activated, right? Well, why do I care about pyruvate kinase? I want to care about up here. Well, you'll see how it works. Pyruvate kinase catalyzes the Big Bang, remember? Meaning that this is very energetically favorable moving downwards. So what's going to happen to the concentration of phosphoenolpyruvate in the cell? It's going to drop, right? If phosphoenolpyruvate concentration drops, what's going to happen to this reaction, the one before it? It's going to pull it, right? It's going to pull that reaction. And when it pulls this reaction, what's going to happen to this reaction? It's going to pull this reaction. So starting everything down here pulls all of these reactions, which is going to decrease the concentration of these products. Delta G equals delta G0 prime plus RT times the natural log of products over reactants. The smaller that ratio is, the more negative that log term is, right? How do I make that ratio as small as possible? By decreasing products and by increasing reactants. And I just told you how we did it. We increased F16 Bp. F16 Bp started pulling the reactions after it, and that decreased the products. That's how the cell gets around that energy barrier. It's really cool, and it's set up thanks to the regulation of the enzymes. This refers to a process I call pushing and pulling. The pulling you see down here. Pulling means removing products. Pushing means increasing reactants. Bang, bang, you've made an unfavorable reaction become favorable. Pretty cool. Questions about that? Okay? Let's talk about fermentation. Okay? Yes, question. The three control, you mean the three enzymes that are controlled? So 3 are regulated by the processes that I've talked about: hexokinase, Pfk, and pyruvate kinase. Okay? And the one reaction that I said was controlled was the G3PDH reaction that was controlled by the level of NAD that was present. I'm not sure if I'm answering your question. Okay. All right. Come see me if that isn't, if you think about it more, okay? Fermentation, all right? Well, most people think of fermentation as the making of alcohol. I told you in class the other day that you are fermenting, and you're not making alcohol. When you go for a run, when you go for exercise, what are you doing? Well, here's what you're doing. You're burning up your ATP like we talked about. If you exercise hard enough, you will exercise and use oxygen faster than your blood supply can deliver it to the tissues. You will exercise faster than your blood supply can deliver oxygen to the tissues. Okay? What does that mean? That means the tissues start running out of oxygen. Now, we'll talk about the electron transport system very shortly, but the electron transport system requires oxygen to convert NADH back to NAD. Cells require oxygen in order to convert NADH back to NAD. When they run out of oxygen, they're in trouble. And why are they in trouble? Well, they're in trouble because of what I just showed you here. They don't have any NAD, this reaction doesn't go. If I'm out exercising, my muscle cells are needing energy, but they've got no oxygen. They need to keep somehow generating energy, and they generate energy by recycling their NADH back to NAD by a different mechanism that doesn't require oxygen. So fermentation is a way of recycling NADH to NAD that does not require oxygen. I'll repeat that. Fermentation is a way of recycling NADH back to NAD that does not require oxygen. Now, the first of those is shown in that slide that I just had on the screen. It's right here. This is what happens in our body when we ferment. Pyruvate is the end product of glycolysis. Okay? The end product of glycolysis. If oxygen is limiting, the cell has an abundance of NADH. We have a lot of substrate right here. Okay? This enzyme lactate dehydrogenase converts pyruvate into lactate, but you'll notice it also converts NADH into NAD because this is a reduction. Okay? And NADH is getting oxidized back to NAD. We just regenerated NAD. To summarize it in a more relevant fashion for you, let me show you this guy right here. Here is the reaction I just showed you, going from pyruvate to lactate. This takes NADH and produces NAD. And where is NAD needed? It's needed in glycolysis for the G3-PDH reaction. And now we see how this reaction down here can feed this reaction up here. Fermentation provides NAD for cells. Okay? And it provided under conditions of no oxygen. For those of you who are making beer, you know when you're making beer, you got to cap everything off, because if you let oxygen into that mix, you're going to make a mess. The reason you're going to make a mess, is I'll show you in a minute, is because you're not making NAD. Okay? You're letting the cell go to do its stuff in a different way. All right, everybody understand why this is important? Okay? What happens in the case of generation of ethanol? Well, it turns out that yeast and bacteria are different from us. Okay? We knew that, right? But they're different from us, and they're different from us in the way that they ferment. Okay? They also have to keep this G3-PDH reaction going, but instead of converting pyruvate to lactate, they don't have lactate dehydrogenase. Instead, they have an enzyme called alcohol dehydrogenase. And what does alcohol dehydrogenase do? Well, it converts acetaldehyde into ethanol. That's the part that makes the stuff for the brew. And it converts NADH back to NAD, just like the lactate dehydrogenase did. Their view of fermentation is different from our view of fermentation. Where'd the acetaldehyde come from? Well, the acetaldehyde came from the oxidation of pyruvate. I shouldn't say oxidation, but the decarboxylation of pyruvate. So they also have pyruvate like we do, but instead of converting pyruvate into lactate, they convert pyruvate into acetaldehyde, and then they do fermentation. Now, I'll tell you something that will surprise you. We have this enzyme right here in our body, alcohol dehydrogenase. Okay? Why don't we make ethanol? Well, the first answer to that question is because we don't have the enzyme that converts pyruvate into acetaldehyde. Okay? At least not enzymatically. All right? We have this enzyme, though. Why do we have this enzyme? Any thoughts? What would we have? What use would we have for that enzyme? What's that? To reverse the reaction. Why would we want to reverse the reaction? We're not into sugar, but you're on the right track. So we have this enzyme to run the reaction from left to right. Ethanol is toxic. Ethanol is toxic. And so going backwards to acetaldehyde is a way of detoxifying, reducing the concentration of ethanol, and we make acetaldehyde. And this is why we have hangovers. Oh, yeah, now it's relevant, yeah. This is why we have hangovers. Okay? Acetaldehyde is very readily oxidized. It's very readily made into acetic acid. And some people argue that acetic acid is actually what causes the hangover. I won't get into that debate, although I'm willing to do the research. You guys are slow today. Okay. All right, does that make sense? All right, so that's what's happening in fermentation. In both cases, whether it's our cells or the yeast cells or the bacterial cells, the aim is making NAD when oxygen is limiting. Okay. Blah, blah, that's just how you make acetaldehyde, or how the yeast makes acetaldehyde. And I won't talk about that. Okay. Okay, that's what I want to say about glycolysis. Now, if you're sitting here going, oh my God, we got another entire pathway before he finishes, there's some good news coming up. Okay. Do you trust me on that or not? No. No. He doesn't trust me. Okay. All right. The good news is if you learn glycolysis, you've learned most of gluconeogenesis, which is the pathway I'm getting ready to show you. Okay? Glycolysis is the breakdown of glucose into pyruvate. All right? Gluconeogenesis is the synthesis of glucose from pyruvate. You might say, oh, we just go backwards. Well, partly. There are 10 reactions in glycolysis. There are 11 reactions in gluconeogenesis. That tells you it's not exactly the same. 7 of the 10 enzymes in glycolysis are the same enzymes in gluconeogenesis. 7 of the 10 are the same enzymes in gluconeogenesis. That means 3 enzymes in glycolysis are substituted for 4 enzymes in gluconeogenesis. Okay, so far so good. The 3 enzymes in glycolysis that don't appear in gluconeogenesis are the 3 that I made you memorize, the regulatory enzymes. Pyruvate kinase is not used in gluconeogenesis. Pfk is not used in gluconeogenesis. And hexokinase is not used in gluconeogenesis. Instead, they have different enzymes that catalyze slightly different reactions, and I'm going to show you those. Everybody with me so far? So the enzymes that are different, okay, so the enzymes of glycolysis that are not used in gluconeogenesis are hexokinase, Pfk, and pyruvate kinase. Gluconeogenesis has different enzymes that catalyze slightly different reactions, and I'm going to show you those. Okay? Anybody think of any common features of those 3 reactions that I told you? Any commonalities in them? There is. They all have ATP connections. Okay? Now, there's only one other reaction in glycolysis that has an ATP connection, and it's not, I mean, it appears in both pathways. But these 3 all have ATP connections. Pyruvate kinase makes ATP in glycolysis. Pfk uses ATP in glycolysis. And hexokinase uses ATP in glycolysis. All right. Well, let's take a look at those reactions. All right? We start doing gluconeogenesis from what I call the bottom and then move upwards, because we've been thinking about glycolysis as going from the top downwards. We're going to start at the bottom and move our way upwards. Okay. Here's the first enzyme of gluconeogenesis. It's called Pyruvate Carboxylase, and yes, you should know the name of it. And this enzyme is found in the mitochondrion. By the way, all the enzymes of glycolysis are found in the cytoplasm. All of the enzymes of glycolysis are found in the cytoplasm of the cell. So here we're starting out gluconeogenesis, and we're starting in the mitochondrion. And we're starting with an enzyme called Pyruvate Carboxylase. And I mentioned earlier in the term the coenzyme known as biotin. And I said whenever you see biotin, you will almost always see that a carboxyl group is being added to something. And if you look, that's exactly what's happening here. We're starting with a molecule of pyruvate, which has 3 carbons, and we're making a molecule of oxaloacetate, which has 4 carbons. We've therefore had to pick up a carbon from carbon dioxide. And we also had to use some energy to make this happen. Okay? We had to use some energy to make this happen. That's the first enzyme of gluconeogenesis. The second enzyme of gluconeogenesis is the very next enzyme. It takes that oxaloacetate that has just been made, okay, and converts it into phosphoenolpyruvate. That was a glycolysis intermediate, right? What did we just do? We just did 2 reactions to bypass the Big Bang. Right? The Big Bang is a very energy-releasing step. If we want to go around a very energy-releasing step, we're going to have to put a lot of energy in. If I jump off of a cliff, okay, and I land at the bottom and I survive, I'm going to have to put a hell of a lot of energy climbing that cliff again to get back up to where I was, right? All right. And I might not go back up the same way I came down, right? I'm not going to fly to the top. I've got to find a way up. That's what the cell is doing. The enzyme that catalyzes this reaction is called Phosphoenolpyruvate Carboxykinase. Big mouthful of a name. You can call it PEPCK. PEPCK. This enzyme's in the cytoplasm. So we've moved immediately from the mitochondrion to the cytoplasm. Notice it took energy here as well. And look at the energy that it took. It took GTP. Now I want you to remember that on that lower half of the Y of glycolysis, we have two molecules of each of these. It takes 2 pyruvates to make 1 glucose, right? So we're starting with 2 pyruvates and making 2 oxaloacetates, and we're taking 2 oxaloacetates and making 2 phosphoenolpyruvates. That means it's going to take 2 GTPs here, and it took 2 ATPs in the first step. Okay. Well, now where are we? Well, we've now made phosphoenolpyruvate, and here's the part that you're going to like. The part that you're going to like is that we're just going to reverse the other reactions. We've made PEP right here. We reverse the reaction to get 2 phosphoenolpyruvate. We reverse that reaction to get 2 phosphoglycerate. Reverse that reaction to get 3 phosphoglycerate. We reverse that reaction to get 1,3-bisphosphoglycerate. In fact, we reverse every reaction all the way up until we get to the PFK reaction. And the PFK reaction is shown right here. And you can see the enzyme that gluconeogenesis uses. It's called fructose 1,6-bisphosphatase. And you can call it fructose, let's call it FBPase. How about that? FBPase. What's that? Yeah. So the enzyme of gluconeogenesis that replaces PFK is called fructose 1,6-bisphosphatase, or you can call it FBPase. Okay? Notice that this is not catalyzing the simple reverse of that reaction. The PFK reaction, if we were to reverse it, would require converting ADP back into ATP, and we're not doing that. Instead, we're simply clipping one of the phosphates off. I told you we're doing slightly different reactions. That's why we're using different enzymes. Okay? The last enzyme of gluconeogenesis that's different from glycolysis is the enzyme called glucose 6-phosphatase. That replaces hexokinase. This enzyme, again, clips a phosphate instead of regenerating the ATP. And this last reaction is the only one besides the first reaction that doesn't occur in the cytoplasm. This guy up above occurs only in the endoplasmic reticulum. Okay. Now let me summarize some things for you. We've started with 2 pyruvates. We've made 1 glucose. Energy-wise, what did that cost us? Well, I didn't add them up going through here, but I'll tell you briefly. Energy-wise, it requires 6 triphosphates to reverse the pathway. 4 of those triphosphates are ATPs, 2 are GTPs. If you want to consider them as 6 ATPs, that's perfectly fine. It also required 2 molecules of NADH going back to NAD. This is an anabolic pathway. And you remember that anabolic pathways start with small molecules, make bigger molecules. They require energy instead of producing energy. And they are reductive in nature, like this NADH reaction going back to NAD. Zunai. I'll stop for a minute. Thank God, right?
You said the last one required 3?
I'm sorry?
It required 3 of those?
3 of what?
The NAD?
No, 2. 2 NADHs produce 2 NADs.
Okay.
Yeah. So you need 6 triphosphates and you need 2 NADHs to go back to glucose. Okay? Everybody stand up and stretch. Let's do that. You guys look like you're dead. Stand up and come on, just stand up and right? Think positive exam thoughts, right? Jumping jacks. Ready? All right. 1, 2, 2. Nobody but me. Okay. All right. You guys look like you have too much acetaldehyde. Is that true? No? Okay. I'm just going to say a couple more things about this. Notice this figure shows glycolysis going down. It shows gluconeogenesis going up. Okay? There's the individual reactions. There's this guy. And this is something I want to talk about. There's the fructose 2,6-bisphosphate I told you about before. Why am I bringing it up now? Okay? Well, this actually, what's that? To make it hard? Is that what you said? Oh, no. This, I think, will simplify this for you a little bit, okay? Hopefully. I said regulation was important, right? Because we don't want cells breaking down things that they might need. We don't want cells making things that they don't need, right? So there's certain times that we want cell to be going through glycolysis, and there's certain times we want a cell to be going through gluconeogenesis, right? When does a cell want to be going through glycolysis? When it's got low energy. Conversely, when does it want to go through gluconeogenesis? When it's got high energy so it can store it. It stores it in the form of glucose. This happens primarily in your liver and your muscles. Primarily in your liver and in your muscles. Okay? The energy needs are completely different, right? One's a low energy, one's high energy. We want the cell to be doing opposite things. We want it to do one thing when it's got low energy, and we want it to do something completely different when it's got high energy. And now we've got a pathway that's got many overlapping enzymes. Right? Many overlapping enzymes. How do you control which way it's going, the glycolysis direction or the gluconeogenesis direction? Well, that's accomplished by controlling those regulatory enzymes. The regulatory enzymes you saw in glycolysis were the very enzymes that were replaced in gluconeogenesis. And it turns out those enzymes in gluconeogenesis are also regulated. F26 Bp activated Pfk. What do you suppose F26 Bp is going to do to FBPase? The opposite effect. It's going to turn it off. Things that turn on glycolysis will turn off gluconeogenesis. Things that turn on gluconeogenesis will turn off glycolysis. This is what we call reciprocal regulation. I'll repeat that. This is what we call reciprocal regulation. It occurs when pathways are regulated in opposite fashions by the same thing. This guy turns PFK on, it turns FBPAs off. It's very simple. Now the cell doesn't have to worry about having both of these pathways going on at the same time. Let's think about what would happen if both of those pathways were going on at the same time. Glycolysis is going on. Okay? How many ATPs do we get out of glycolysis? We get a net of 2. And gluconeogenesis is going on. How many triphosphates does it take for gluconeogenesis? 6. If both are going on, here's what's happening. Glucose goes to pyruvate, makes 2 ATPs. Pyruvate goes to glucose, uses 6 triphosphates. We've gone in a circle, and we've had a net loss of 4 triphosphates. If that circle keeps going on and on and on, what's going to happen in the cell? Well, the cell is going to be burning triphosphates, and it's going to be making absolutely nothing, because it's going back to the very thing it started with, which is glucose. This is an example of what we call a futile cycle, F-U-T-I-L-E. A futile cycle occurs when catabolic and anabolic pathways are occurring at the same time. That's what a futile cycle is all about. And cells really, really, really avoid futile cycles. There are some cells that like futile cycles. Wait a minute, you just said, Kevin, that cells avoid futile cycles, and now you say some cells like futile cycles. Why might a cell like a futile cycle? Anybody have any idea? What's a futile cycle generating? If it's not generating a molecule, what would it be generating by going in that cycle? Heat. Heat. Okay? Futile cycles can be very beneficial for generating heat. It's the one thing that they do that's of any use to a cell. Okay. You know what I'm going to do? I'm going to leave Cori cycle to the next time. Cori cycle is a very simple cycle, but you guys look tired. I think I'm going to sing a song to you to plant gluconeogenesis in your heads. Is to the tune of supercalifragilisticexpialidocious. Okay? And it's right here. When cells have lots of ATP and NADH2, they strive to store this energy as sugar. Yes, they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD, and G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose factory, glucose factory. The aldolase reaction puts together pieces so. A fructose molecule is made with 2 phosphates in tow, and one of these gets cleaved off with a fructose phosphatase, unless F26 Bp's acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose jubilee, glucose jubilee. From F6P to G6P, that is the final phase. The enzyme catalyzing it is anisomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmical reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard because our professor lets us use a filled out index card. Gluconeogenesis is not so very hard. Okay.